Okay, here we go with part 2 of our lesson on energy of membrane transport. And so in this particular video, we're going to look at the concept of free energy as it applies to symports and antiports. And then we're going to look at detail of the sodium-potassium pump. So to kind of recall from last time, antiporter has two ions or molecules going in opposite directions, one coming in and one going out. So that's what we're looking at over here on the left. Now it could be that one of these species is going in the direction of the gradient and the other against, or they could both be going against the gradient and it could be facilitated by some sort of other reaction. Often that's the hydrolysis of ATP, and we're going to look at that example with the sodium-potassium pump. In a symporter, both molecules or ions are going in the same direction, and usually one is going with the gradient and helping push the other one against the gradient. Okay, let's look at this example. Our goal is to figure out if this sodium glucose symport, so symport, they're both moving in the same direction. We see that here with the arrows. They're both moving into the cell. Would this work spontaneously or would it require energy from ATP? So let's look at things one at a time. As we look at the sodium, I see it's going from an area of higher concentration to an area of lower concentration. And so we know that that is favorable. Okay, the concentration, let's write that, sodium, the concentration gradient is favorable. And if I look at the charge, let's come down here and look. Our membrane potential is negative 0.05. We know if it's negative, that means there's positive charge on the outside, negative on the inside. And if I come back and think about sodium, sodium is positively charged. It makes sense that it would move towards that negative charge, and so the membrane potential is favorable also. So I would expect that for the sodium ion, that is going to have a negative free energy change. It's going to be a spontaneous reaction for sodium to move in. Now let's think about the glucose. So glucose is going from 4 millimolar to 40. That is not favorable, right? So for the glucose, whoops, not what I wanted. For the glucose, the concentration gradient is unfavorable. And when we look at the charge, glucose really isn't a charged molecule, so we're not going to worry about that part as much. Glucose travels in and out of cells quite a bit. It's the primary energy source in animals. And this is a particular case where we're going to have to somehow push the glucose into the cell if we need it there, because we know that that piece of it is not favorable. So to figure out if this would be favorable, we are going to need to calculate the change in Gibbs free energy for each of those transfers. We know the sodium will definitely be negative, and we can calculate that value. So remember our equation down here. Whoops. And so for sodium, if I'm going to calculate that, so the change in the total Gibbs free energy for the sodium ion, we know it's going to be that R times T, 8.315. I'm going to leave off my units, which is a horrible example, except we did those clearly in the last video, and it will give us a little bit more space. We are going to assume that we're at our same, we're not assuming, we are told we're at 37 degrees, so we've got 310 Kelvin. And then natural log, and I know C2 over C1 is the final over the initial. And so my final, it's moving into the cell. So at the end, it'll be in this area that was lower. My initial is here. So that's the contribution from the concentration gradient. And then I have to take into account the membrane potential. Sodium has a +1 charge. I know Faraday's constant. And I know the voltage that was given. Whoops, and that was my cat helping us. There we go. I can do this same thing for glucose. So calculating the total Gibbs free energy change for the glucose. The same starting point. But now my final concentration, we're in that 40, here is 4. And then if I think about this last piece for glucose, the charge on glucose is 0. So that whole term, this whole term right here, goes away. So I don't even have to worry about that piece. If you come through here and you tally up these two, right, the total from the sodium plus the total from the glucose, we find it is negative 5.4 kilojoules per mole. So this would indeed be spontaneous. So this symport essentially has the sodium ion carrying the other species across that gradient. All right, now we want to talk about the idea of secondary transport. Because if we think about that sodium glucose transport that we just looked at, so that's shown over here on the right with this symport, it works because the sodium gradient was already set up such that there was a lot more sodium on the outside, right? We had a high concentration of sodium on the outside, so it would naturally flow into the cell. If that concentration gradient hadn't been there, this symport would not work. And so this is considered a secondary transport because something else had to set up that concentration gradient to start at first. And so over here, this piece was actually our primary transport. Okay, it's setting up that concentration gradient. In this case, it's that sodium-potassium pump. And we're going to look at that in depth. Notice that when I look at this, there is a video here. So if you want to look at a picture of this, you can play this video. All right, and the video calls it cotransport, but also symport is the same thing. All right, let's look at that sodium-potassium pump more in depth. It is an example of an antiporter. Every time this happens, for every cycle, we get 1 ATP hydrolyzed. We get 3 sodiums moving out of the cell, pumped out, and we get 2 potassiums pumped into the cell. And in this case, both of these are going against the concentration gradient. So it's unfavorable, so that ATP must be fueling this. And let's look at how this would happen. This reaction shows the net, right here, the net change. So we see we start with potassium on the outside of the cell, and we end up with potassium on the inside of the cell. We start with sodium on the inside, we end up with sodium on the outside, and then we have this ATP being hydrolyzed into ADP and the phosphate. The net reaction is spontaneous. So we know that the sum of all the different energy changes for all of those processes must give us a negative free energy. And that means, since we know both the potassium, the one underlined in blue, and the sodium, the one underlined in orange, since we know those are both going against the concentration gradient and they would have positive delta G values, this delta G value must be pretty negative. So let's look at how we get that energy from ATP. This a little bigger. And actually, if we look here, so at the top we have our ATP. So right here. And this reaction series is actually showing two pieces. The part we care about right now is just that first step, going to ADP. But this was just interesting, so I left it in. This reaction can continue to form this AMP. And this is a nucleotide found in RNA. And actually, if you sub out this for a hydrogen, now it's nucleotide found in DNA. And so this is part of the beauty of the biochemistry, that these important molecules, starting with the ATP up here, can be used for many different things as we go through. But for now, we care really about this top part. So let's take a look at that. Just looking at the first step, we have ATP. We have water coming in to hydrolyze the phosphate. So we see right here, this oxygen attacks that phosphate. That breaks this bond right here and gives away our phosphate. So we get that inorganic phosphate. And if we calculate the standard Gibbs free energy for this reaction, that's a lot. Negative 32 kilojoules per mole is quite a bit of energy. And our question should be, well, why? Why is there a high release of energy? And there's actually several reasons. First of all, take a look up here along the bottom end of this triphosphate. You've got lots and lots and lots of negative charge. And we know that like charges repel. So when we break apart that one phosphate, we're moving some of those negative charges away. And that lowers the energy of the system. Okay, so it stabilizes the system. So one reason is, let's see, number one, separation of some of the negative charges. A second thing we can look at is the fact that we started with one ATP molecule and we're forming two molecules. So the entropy factor plays a role here. If you remember, delta G equals delta H minus T delta S. Since our entropy is increasing, that is contributing to making delta G more negative. The water doesn't really count because it's really part of our solvent. So we've got one molecule going to two molecules. And then a third thing we can look at is this phosphate. When the phosphate is separated, there is more resonance, which is stabilizing. So stabilizing, lower energy, so some of that energy is given off. So there's several things going on here to give us a lot of energy from this ATP. If we think about that in terms of our reaction and our numbers, the standard Gibbs free energy change we just said was negative 32 kilojoules per mole. But we know that in cells, the concentration of ATP is typically greater than the concentration of ADP. Remember, this reaction was ATP goes to ADP plus that phosphate. So Q, right here, this is Q, is products over reactants. So products over reactants. So this number, Q, is going to be less than 1, making this whole thing negative, so contributing to delta G being even more negative. And so when we look at that, it turns out that delta G for the reaction is in the range of about 50 negative 50 kilojoules per mole. So that's a lot of energy, and it turns out it's enough to push those sodiums and those potassiums across those concentration gradients. And then the question you should be asking yourself is, okay, but how? How can we harness this energy from ATP to physically get these sodiums and potassiums to move across this membrane? There's our question. And we're going to take a look at that. As part of this, we're going to look at this phosphoryl aspartate. So we know that we have ATP, you're going to get tired of me writing this, aren't you? ADP plus the phosphate. And the reason this can happen is because in this process, there's an intermediate in which the enzyme, okay, the enzyme that's this pump that's pushing the sodiums and potassiums in and out of the cell, is phosphorylated. Okay, so these types of pumps are called P-type ATPases. So the ATPase is the enzyme here, and this is P-type because it is bound to this phosphorus in the intermediate. Let's take a look at what that looks like. So first let's look over here on the left at this kind of cartoonish structure. If we look, it has multiple subunits. So each of those different colors and different pieces is a different subunit. Okay, the green ones are the transmembrane subunits. Okay, and then if you look down at these, you've got the ones that are down in the cytosol. So here's the inside. Let's make sure we label that. So this is the inside of the cell, and here's the outside of the cell. If we look over onto the right, oh, and let me point out on this blue piece right here, somewhere on that blue piece is the ATP binding site. It's going to turn out that that binding is going to cause a conformational change that then allows these sodiums or potassiums to move in and out. Okay, so ATP binds here, and the phosphate is passed on and off also, so that free phosphate can also bind somewhere on that blue one. If we look over here, you see the ribbon drawing. You'll see we have those alpha helice motif crossing the membrane in that transmembrane portion. Okay, and somewhere down here is where we expect that ATP and phosphate to bind. All right, so now we have a bunch of all right, sorry. Outside noise distracting me. All right, here we've got these pictures. And now my cat's biting me. Y'all, it's not my day to make videos, evidently. Picture of this cycle. We are going to start down here in the middle. You could start anywhere you wanted, but I want to start right here. When I look at this, I'm going to identify that this yellow part is my ATP. And so my ATP is bonded to the binding site. So let's say in number 1 here, ATP binds to active site or enzyme let's just say to the enzyme. Okay, remember that binding's reversible. It can go on and off. And when it binds, it causes a conformational change, allowing those sodium ions to enter. Okay, so here we're going to see those sodium ions, those 3 blue dots, having entered right there. Once the sodiums have entered the pump, if we look at what's happening over here, the inorganic phosphate is staying attached, but the ADP is leaving. Okay, so the phosphate stays bound. It's bound to the aspartate. Remember, we looked at that intermediate. And the ATP is released. And when this happens, it causes a conformational change that opens the plasma side. So it closes the cytosol side and opens the plasma side. And at this point, when that happens, right up here, so if we think about this as step 2, where that ATP breaks apart into ADP and phosphate, once that happens, that conformational change allows the sodium ions to leave. Okay. And as that is open, we see that the potassium ions enter. And if you think about why that might happen, these conformation changes, when the enzyme is open to the cytosol, like it is down here, okay, the conformation is more favorable for sodium ion to bind. But when it opens up to the plasma, something about that conformation change makes it less favorable to the sodiums, so they float away, but it makes it more favorable for the potassiums, so they enter and bind into that site. Okay. Then as we move across here, notice that we've got those potassiums in our binding site. And then look at what is happening here. We have another ATP bonding. As it does, that phosphate leaves. And that ATP binding causes another conformational change, right? And so we see that right here. So we can, let's just keep our list up here. So another ATP binds. Which then opens, changes the conformation, right? And it opens the side to the cytosol, opens the inside, and releases those potassium ions. Okay, for each cycle that this happens, we have one ATP getting converted to ADP and phosphate. We've got 3 sodiums going out and 2 potassiums coming in. This video is a picture of that, so you can take a look. If you watch the yellow kind of star-shaped thing is your ATP, and you can watch it break apart into the phosphate and the ADP. Useful to watch. All right, let's try a calculation for this sodium-potassium pump. We want to calculate delta G for one cycle of this pump. We have already been given the idea, or given the value, that we expect the ATP piece of this going to ADP plus the phosphate. Delta G should be around, I keep forgetting the negative, y'all, but we know it's giving off energy. So what you need to do is calculate the total Gibbs free energy change for the sodium and for the potassium. We've done this before earlier in this video, so I'm not going to take the time to do it, but I do want to point out that when you do this calculation, when you calculate, say, delta G total for the sodium, you have done it for one sodium. So you're still going to plug in because you're doing the charge on each ion. You're still going to plug in +1 there. So you're doing this for one sodium. And we know we're moving 3 sodiums. So once you do that, you're going to have to multiply the number by 3 to get the total Gibbs free energy change for all 3 sodiums. And when you do the same thing for the potassium, you're going to have to multiply it by 2. When you add all of those things together, you should find that the energy needed to run the pump should be about 43 kilojoules per mole. And we saw that we get plenty there. All right, next, this is a video on how neurons work. This is really outside the scope of the course. We're going to talk just a little bit about the piece of it we care about, but you could watch the video if you're interested. And here's your video. And what I want to point out is just the idea that these membrane charges are really important for many reasons. They affect the spontaneity of ions moving in and out. If we think about a cell membrane along an axon, so we've got something like this. We've got on the inner side a bunch of negative charge. We've got on the outer side a bunch of positive charge. And we've got somewhere our sodium ion pump that we just looked at, right? And that's keeping this going. We've got the right concentrations of sodium on the inside, the right concentrations of potassium on the outside. If you watch that video, what you'll find is that when some neurotransmitter, some chemical like something disrupts this, okay, and so maybe for a little bit, this is backwards and positive, 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 negative, negative, negative. This will open up some other channels for sodium to flow in or out, right? Because if you've got a ton of sodium on one side of this, then it's going to, when you open a channel, it's going to naturally flow towards that negative charge, right? Or a channel for potassium. And what happens is we have all these channels, and this charge was disrupted, and so your ions move until it resets the charge. And this propagates down. You can watch the video, but it propagates down the nerve cell. And so you get this changing of the charge to propagate down. And the piece I really want to look at, you can watch the video if you want to look at the propagation piece, but the piece that will make sense to us in our scope is right here. Here is a channel that would allow potassium to enter or leave the cell. Okay, we know that naturally it would normally go towards the lower concentration area. This channel has all these alpha helices. It's actually dimers, so the left and the right side are the same. And I really want to highlight that on the alpha helices that we've labeled with 4, that these things are positively charged. Okay, and they're positively charged because they are mostly lysine. And so that primary structure is mattering here because now we have these positively charged side chains. Well, if this is the inside of our cell, we know we've got a lot of negative charge. If this is the outside, we know we've got positive charge. So if these ones shown in blue are positively charged, they get pulled down and it keeps this gate in the closed position. But when this positive, negative cell potential is disrupted, somewhere along the way. And so now we get, whoops, now we have negative out here, positive in here. Suddenly, these positively charged alpha helices are pulled up, and that opens up this channel so the potassiums can flow out, and they'll naturally flow up towards here. And over time, that'll disrupt this, right? You're adding more and more positive charge, and so then we'll close the gate again. So that's just an example of how we can use what we know about the amino acids. And our whole goal here is to connect the conformation changes, the chemical structure with the conformation changes with the function of the molecule. All right, that was a lot. That was energy of membrane transport, and that is it for this video.